Hello, Eastview Scholars. Welcome to this edition of Sharp Shorts. When last we talked in our last tutorial video, we were looking at meiosis and how it's a type of cell division that leads to genetic variability and as part of sexual reproduction in the fusion of gametes. We also looked at asexual reproduction as a process that most organisms outside animals can use, also based on environment, life cycles, etc., etc. When we were looking at the benefit that sexual reproduction brings through the fusion of gametes, we were essentially looking at animal cells, this life cycle here, where, as you can see with all these different sexual life cycles, meiosis and fertilization are the key to that sexual reproduction and the variety that it brings in genetic organisms to make those offspring not quite the parents, not the exact cloning that mitosis and asexual reproduction brings. And so what we looked at is in that life cycle, all life cycles, we should say, start from the fusion of the gametes and the formation of that zygote until that organism then develops to maturation and reproduces on its own. In animals, as we see here, the meiosis is used by the diploid organism to create the gametes, and then the gametes get fused, go through fertilization to create a diploid zygote, which then goes through mitosis to create the fully grown animal, and that is our life cycle. Now, if you look at on the other side, and I'll skip over here to most fungi, I'm a fungi, and some protists, what you can see here is essentially it's an opposite. You have those organisms spend their life cycle, most of their life cycle, in a haploid state, not a diploid state. And the only time that they are diploid is in that fertilization and short zygote stage, as opposed to our very short stage and long diploid stage. So what happens then is the zygote actually goes through meiosis, if we want to follow the same thing. The meiosis is done by the zygote to create a haploid organism. And that organism then goes through mitosis to get to its life cell, and then it's the fusion of the gametes of those that create the fertilization to create the zygote, which then immediately goes through meiosis to be the haploid organism. So you can see just by looking at the amount of time spent in the life cycle that we spend our life cycle mainly as a diploid organism, only using meiosis to form gametes, where fungi and some protists spend their life cycle haploid and only use meiosis to become that haploid organism, mitosis to grow, and then the fertilization is only to have that zygote moment. Plants and some algae really are kind of halfway in between. As you can see, circles are a good idea. They go through what is called alternation of generations. So they live as organisms in both a haploid and a diploid state. Starting here again, what you can see is the diploid plant is called a sporophyte, and what it has done after fertilization has grown through mitosis and created spores, or has a sporophyte, a spore pod there and stuff like that. And then what happens is that organism then goes through those spores, goes through meiosis to create those spores. Those spores are then released, and through mitosis, they make the haploid multicellular organism that's called the gametophyte. And so they have that life cycle or half of that life as a gametophyte in one generation, and then those gametophytes from the spores produce the gametes fuse to fertilize the zygote, and then their that generation then is the sporophyte stage, where they produce the spores Through meiosis, the spores are released. Through mitosis, they form the gametophyte and so on. So you can see that different organisms, it's in the timing of the meiosis and the fertilization. Okay? Now, so behavior of chromosomes during meiosis and fertilization is responsible for most of the variation. So the three that we're going to look at quickly, where does that variety come in? If the biggest thing about sexual reproduction is the ability to adapt in that competition and to have genetic variation, so you're not a clone of your parents and you can adapt to different environments and stuff along that line, what happens? Well, there's three things. Independent assortment of chromosomes, which we'll talk about, which is when those homologs are lining up in meiosis 1, it doesn't matter the order that they do. Crossing over, we looked at meiosis is when those homologous chromosomes are close to each other and the tetrad is formed, that there are parts that swap chromosomes, swap genes, sections of the chromosome, and random fertilization is, any male, any sperm can fertilize any ovum in a sense. So let's look at them in a little more detail. Here's independent assortment, and this is just a two chromosome organism. You can see that the homologs are paired up and stuff. Now, which side is mom's, which side is dad's, doesn't matter. Okay, so you could have, say, both dads on the left, both moms on the right, and they split that way, and not assuming any of the crossing over or anything here, then you get organisms like such. Or dad can be here and mom can be here and mom can be here and dad can be here, and then you get in your 4 haploid cells a different combination. Okay, that's independent assortment. How the homologs, chromosome 1, line up is completely different, mom, dad, than how the homologs for 2 or 3 or more. Okay, so in general, what are the combinations of those different chromosomes lining up independently, mom on one side, dad on the other, or whatever it is? It's 2 to the n being the haploid number. So if you think about it, what is the possible combinations for human? That's 2, the haploid number is 23, that's 2 to the 23rd. That's about 8.5 million combinations of the homologs lining up in different ways. And that's just one parent. That's not both parents before crossing over. So you can see that just in that independent assortment, what goes into that gamete and then what may possibly be the combination from the gamete that you get along with dad's and mom's together is a lot. Really what it leads to before even a crossing over with 2 parents is about 70 trillion combinations. So the odds that you're going to get one very similar to your parents or exactly like your parents is very slim. Okay, so just that idea. Crossing over we looked at, so I'll just go through this quickly. Here you have at synapse the homologs. Synapse, and what you do is you get that bonding with what are called cohesion proteins and stuff, and you can get crossing over at these chiasmata where they're going to swap, and they do it very well here with colors. They're going to swap those chromosomes. So at metaphase, when you separate the homologs, you have separated. You can see now by the time you go through anaphase and you're lining up on metaphase 2, these do not look like the original. And so you get some recombinant chromosomes here. That leads to genetic variation. And that's that's added on to the 70 trillion combinations of organisms that a human zygote could get from their 2 parents. Okay? The last thing is random fertilization, and that adds to variation because basically any sperm can fuse with any ovum. Okay, and we talked about this goes with the number. You get 8.4 million possible chromosome combinations from independent assortment from each parent. That means 70 trillion diploid combinations, one from each parent. That's before the crossing over. That's just a very strong formation variation that allows organisms to be carry characteristics of their parents, but be adaptable to the environment on your own. Hopefully that gives you an understanding of the genetic variation that sexual reproduction can bring. Scholar up, Eastview.